hopefully you can see this. So obviously I come from University of Birmingham. We're one of the biggest universities in UK, and we're one of the biggest medical schools in the country as well. Let me just put a laser pointer. There we go. So this story actually started by me seeing, meeting Matt for the first time at the ISHR meeting, I would say, I think it was in Argentina, in Buenos Aires. And he actually was presenting his work and I just thought it was such an incredible idea. And then we helped them out, get it to a public, take it out to the publication stage in circulation research. But essentially the idea is about a new way for isolating cardiac myocytes does not rely on a Langendorff methodology. But if we go back in time, the first paper, to our knowledge at least, which utilized a Langendorff methodology to isolate and dissociate cardiac myocytes from a rat heart, was published in 1970, again in circulation research, by Michael Berry. And really since then, there's been quite a strong expansion in the use of isolated mouse cardiac myocytes for the purposes of cardiovascular research. So this is a number of publications that contain the words mouse cardiac myocytes on PubMed, and you can see that there is an exponential growth here, and it's actually plateauing a little bit in the recent years. And hopefully that plateau is not instigated by our publication. So Matt really identified that there are several problems with the Langendorff isolations. I mean, whilst it's being used successfully in many labs, you do require a commercial and custom-made apparatus, which is expensive. So for somebody starting out fresh, this is quite a substantial investment. Probably more importantly is that you require considerable expertise. Mouse aorta is very small, and cannulating it successfully is actually quite a tricky thing to do. Of course, there are some other technical issues. You often require pre-injection of animals with anticoagulants, such as heparin, which can affect downstream PCR-based analysis. And actually, probably importantly as well in all this, is there are issues with sterility. Keeping that Langendorff apparatus sterile is not an easy feat. And I think we've all encountered moments where there are weeks and weeks that we can't get cardiac myocytes, and then you sterilize your apparatus with anything under the sun, and then all of a sudden, miraculously, it starts working. So the principle of this approach is based on clamping the aorta. So if we have this is a beautiful illustration that Matt, I think, has produced. And if you clamp the aorta here, then you actually inject your buffers that contain collagenase, for example, into the ventricle. And then that way, the only way they can go is actually through the coronary circulation, and then they distribute the enzyme through the coronary circulation. So effectively, the three key modifications that Matt actually came up with is that the hearts are rapidly perfused with high EDTA buffer. This is to quench calcium and therefore prevent cell death, presumably, but also to destabilize the extracellular matrix. pH of the buffers was adjusted to 7.8, and all the buffers obviously are introduced via this intraventricular injection rather than through a Langendorff system. So a quick run through of a step-by-step guide to the injection method is that you open up the chest in an anesthetized mouse, just below the diaphragm. You identify the descending aorta and inferior vena cava, and then you cut them, and then you inject the EDTA buffers into the apex of the right ventricle. You essentially flush out the heart that way from the blood. You also stop the contractions, and then you reach around and you use the Lahey forceps to clamp the aorta, and then you remove the heart behind the clamp. Then that heart is exposed to more EDTA buffer through the left ventricle, through collagenase buffer, of course, through the left ventricle. And then it follows the normal process of heart being separated into respective chambers, tissue pulled apart, triturated, and dissociate essentially by pipetting. And again, as everybody else knows, then we just let the cardiac myocytes sink to the bottom. The non-cardiac myocyte fraction is often in the supernatant, and that leads to a method, effective methodology, which produces high yields of live rod-shaped live yields of live rod-shaped cardiac myocytes. We've successfully isolated ventricular as well as atrial cardiac myocytes, and the cells that hopefully most of the time look as beautiful as in these photos. We've robustly investigated in the publication in 2016 in Circulation Research, and we've identified that these cardiac myocytes do retain all the transcriptional and functional characteristics. So we've basically robustly examined and demonstrated that they retain the expected frequency-dependent changes in calcium transients. They can be patch clamped and are also amenable to adenoviral transfections. They're responsive to adrenergic stimulation in a dose-dependent manner. They are responsive to pressure overload. In other words, we see cardiomyocyte hypertrophy with increased expression of markers such as AMP, BMP, alpha-actin, and also they're responsive to hypoxic stress. So I'm not going to go into any of the details for the purposes of this presentation. Feel free, please, to look at a paper that was published in 2016 in Circulation Research. What we also demonstrated is that the non-myocytes in the heart represent a heterogeneous population. And when they start being cultured away in the absence of cardiac myocytes, particularly in post-confluent cultures, and a kind of an interesting observation that we've made is that they start developing these beautiful endothelial cell networks. So that you can see here, and obviously they're overlaid on top of all the cardiac fibroblasts as they are stained by vimentin. And that obviously opens an opportunity to study co-cultures of non-cardiac myocytes from the heart, but also co-cultures of cardiac myocytes and cardiac fibroblasts. So in these images here, we show cardiac myocytes stained with cardiac alpha-actinin, and also co-cultured with cardiac fibroblasts stained with vimentin. And these are cell fractions that have been recombined after 3 days of separate cultures. In the process, Matt has done a lot of work on identifying which culture media and which pH are best used in order to allow culture or long-term culture. And whilst there isn't really, so in this graph you can see we're looking at viability, essentially total rod-shaped numbers from day 0 all the way to day 4, and there is a steady decline in the numbers of rod-shaped cells, as you'd expect, but the M199 basal media probably supports the rod-shaped morphology of the cardiac myocytes best. Matt's also investigated the use of different pHs, in other words, culturing at different pHs. And I should just point out that this is done in the presence of FBS, so in the presence of serum, 10% serum. And he's actually noticed that this rod-shaped morphology is best preserved at pHs of 6.7 rather than pH 7.4, where you start seeing this quite consistent remodeling. And I should emphasize that this is obviously present in the presence of serum. In the absence of serum, this remodeling is not as apparent always. And then another interesting observation, which really actually blew my mind, is that if you culture these cells, and this is now in the absence of serum, for let's say 7 days, at pH 6.7 or at pH 7.4, and then stain them with the ethidium homodimer dye, which is essentially a dye that is a marker of dead cells, in other words, gets into the cells and stains dead cells, whereas it doesn't get into the live cells. I mean, this is possibly a little bit difficult to follow, but actually, if you have a look, the cardiac myocytes that are effectively rod-shaped are being stained with this particular dye, whereas the cells that are potentially having these perhaps more rounded structures, the dye doesn't get into them as much. So really this suggests that there is a real distinction between remodeling and viability. In other words, rod-shaped morphology does not always equate with a myocyte viability. And it's something for us to really think about when we're doing, let's say, protein expression studies or RT-PCR in cells that have been cultured. Another really interesting observation that we've come across is that, as we all know, when you culture cells in the absence of serum, these cells will start losing sarcomeric structures and effectively start shrinking. But then if you add, so this is at day 8 in serum-free culture conditions, but then when you start culturing for longer periods, in other words, you start adding FBS, 10% FBS, you really start regaining sarcomeric structures, and also these cardiac myocytes start developing emerging pseudopodia. And I mean, Matt perhaps can help me out here later on, but from what I understand, they also start developing some contractile automaticity as well. So these are, we're not saying these are important for functional studies that we do, but they're definitely important to highlight that these cells have a lot of plasticity in them. And actually, differentiation and development can go both ways, even in the adult stage. So those are the things that really kind of interested me. So when the publication came out, it was followed by an editorial by Chen in Circulation Research, and it was a very fair editorial. It raised some very valid questions that we are now trying to address. And some of those questions are, first of all, will the method be tested with time? And 50 or 100 publications later, 3 years later, we are now fairly certain that this method has been tested for time and other people have been able to replicate it and attain viable cardiac myocytes with it. But another couple of things that they've raised is whether the method would be effective in isolating cardiac myocytes from diseased hearts, such as infarcted hearts, and also that we did no comparisons between the new method and the old method in terms of functional and signaling networks. Now, I'm acutely aware of the time, so I will quickly skip through some of the new data that we will show today. So we have applied this methodology to isolation of cardiac myocytes from infarcted hearts. So myocardial infarction was induced by left coronary artery ligation in 8 weeks of age animals. So these are not large infarcts, as you can see, but effectively we could still isolate cells from the MI hearts. Obviously, the total cell numbers are lower compared to the sham, and the percentage of rod-shaped cells is lower. So that addresses the first point from the editorial. And then we also isolated cells using Langendorff and the injection method and directly compared, so you can hopefully see these are Langendorff, these are injected cells isolated through the injection method. We've looked at diastolic calcium, which was very similar. We've looked at sarcomere length, which is very similar at baseline. We've looked at some of the signaling cascades. So we looked at PKC alpha localization, a very similar distribution between the cytosol and the membrane. We've also looked at ERK1/2 phosphorylation and total expression, again, very similar phosphorylation levels between the Langendorff and injection method. We've also got some functional data. So we've looked at the frequency-dependent changes in calcium transients and contractility in ventricular cells. So we basically pace the cells at 1 Hertz, 2 Hertz, 3 Hertz, and then we go back. These are the calcium transients. These are the sarcomere shortening data. And we looked at diastolic calcium differences at all 3 pacing frequencies, tau at all 3 pacing frequencies, amplitude at all 3 pacing frequencies, and sarcomeric length. We didn't identify any differences between the 2 methodologies, apart from the expected frequency-dependent changes. Finally, we've looked at the response to isoprenolone stimulation. And so we pace at 2 Hertz and then add 1 micromole of isoprenolone. In both Langendorff and injection method, we see decreases in RT50, relaxation time 50, or time to relaxation 50%, with both methodologies, and we see a very similar time-dependent response in sarcomere length shortening and calcium transient amplitude following exposure to isoprenolone. So to really wrap up, we've developed a novel cardiac myocyte isolation method, particularly for mouse cardiac myocyte isolation. It produces high yields of ventricular and atrial cardiac myocytes. It allows co-culture experiments, for example, cardiac myocytes and cardiac fibroblasts. Successful isolation of cardiac myocytes from infarcted hearts has been achieved. And we see no differences in activation of signaling cascades when we compare it to a standard Langendorff isolation. We see no differences in calcium transit and contractility, frequency-dependent responses, and we see no differences in responses to isoprenolone. So, of course, I mean, we have a large team, at least in Birmingham, but also in Singapore. And in Birmingham, the two people that were crucial to this work were Andrew Holmes, who is now actually a lecturer in another institute within the University of Birmingham, and also my previous PhD student, Sean Marie O'Brien, who's actually now in the Navy. So she's escaped science, but she is a very, very talented patch clamper. So I'd like to thank the funders, of course, and I'd like to now stop sharing my screen and give the baton to Matt.
Great. So just check this. Hi guys, so I think we're not going to do questions now, right? We'll leave them all to the end. Is that the best way? So I'll just shoot straight ahead. Can you hear me? Okay, great. I'll get on. Just say thanks to Nina for the amazing introduction, and thank you to Davor for your introduction to our work. It's really been a team effort, and it's been a pleasure to work with Davor and everybody else who's worked together on this project. So I'm going to keep going now, and what I'll do is I'll talk to you about kind of next stage that we've been heading in terms of this protocol, which is more towards isolation of myocytes from neonatal hearts, and also trying to build hypertrophy models with them. So this is me. I'm Matt. I'm working in Singapore at the National University of Singapore, and also the Genome Institute of Singapore. So Davor suggested I should show you exactly where I am. So here's a map of the world, and here's Singapore. So that's where I am. We're right down near the equator. It's a lovely island. We have 4 medical schools. Importantly, some great beaches and great food, and a lot of culture. So I hope as soon as the virus situation blows over, if you haven't been to visit already, then you must come by. So you've heard now about this idea of isolating cardiac myocytes and non-myocytes from not just healthy, but also diseased, and we've done it with aged hearts using our Langendorff free injection protocol, and hopefully you're convinced now that these myocytes are pretty comparable between the two protocols. And the myocytes we've shown, these are adult myocytes. They're amenable to both short and long-term culture and study. So yeah, here's what I'll talk about. So rather than aged and injured hearts, can we kind of downscale this injection protocol now and apply it to neonatal cardiomyocyte isolation in mice? And if we can we build from this a stable in vitro mouse cardiomyocyte hypertrophy model, which is something that's really never been done before? So I'll go back on some history again, as Davor did, but I'm going even 10 years further back. So we're into the early 60s now. So these two papers from the same authors came out together. So really seminal work. And what they showed for the, really, as far as I know, the first time, is isolation of viable myocytes. These are from neonatal rat hearts. And they showed they could culture these cells, and they have a few pictures of them here. And some of the really interesting things is that the observations they made then are still relevant today. So, for example, they observed that these cells, they have spontaneous beating rates, which are different between different cells. But once confluent monolayers start to be formed, you will then see beating in synchrony. So fast forward another 20 years from then. So this is where the real pioneering work from Paul Simpson and McGrath and Savion came in the early 1980s. So I think this was in 1982. And this was a group that really established the model of hypertrophy in neonatal rat cardiomyocytes. So for the first time, they put these cardiomyocytes into culture, and they showed that by using either serum or even more importantly, perhaps, adrenergic stimulation, so in this case, norepinephrine, these cells will both grow in size, so you can see their surface area increases in this 2D culture, and also they increase their protein expression. So these are two markers of hypertrophy, and ever since then, so what are we now? So this is 40 years ago. So in the last 40 years, this has really set the stage for really the majority of in vitro primary culture cardiomyocyte hypertrophy models that around the world most people have been using now. So that again, of course, is 40 years ago, and one of the interesting questions then is why are we not using mice to do this? Because mice are obviously more commonly available in labs. They're usually more accessible to reach and present in greater numbers all around the world. And not only that, but really we know more about mice. Their genomes and epigenomes are better characterized and annotated. Most of our genetic models, including in cardiovascular field, are in mice and not rats. And also there's suggestions that the behavior of myocytes in mice, particularly the adrenergic stimulation and response behavior, is it maybe a bit more similar to humans than rat myocytes. So for example, rat myocytes have 5 to 8-fold more expression of alpha adrenergic receptors than other species do. So in light of these things, you would think maybe that mouse cardiomyocytes were the better model, but I think there's really been two main barriers that have impeded the development of these models. That's firstly the increased difficulty in isolating myocytes from mice, particularly neonatal myocytes from mice, compared to rats. And two is, even when you have these myocytes, they seem, at least previously, to have been less responsive to hypertrophic stimulation in culture, if responsive at all. So I'll address these in turn. So firstly, talking about the difficulties of isolating myocytes from neonatal mice. If you compare this to neonatal rats, it's really relatively easy to isolate myocytes from neonatal rats. You can take these hearts and mince them, and you can do a collagenase digestion anywhere from a few minutes up to 1 hour, depending on your protocol, in a collagenase containing buffer, and you'll yield from that, 1 million, sometimes even up to 5 million reported cells, myocytes per heart. The trouble is, if you take this approach with mouse neonatal hearts, your yields will be very poor. It doesn't seem to work. So, of course, different groups have proposed alternative methods to try and address this, including some which have proposed overnight digestion through the 2-day protocols, using, for example, trypsin to digest in a cold environment overnight, followed by an hour or so of collagenase digestion the next day. Of course, this is a bit complex and takes time. There's commercial kits which are available, if you want to spend that money. And I had to show this. I found there's a group who even have used Langendorff perfusion on these neonatal hearts. So this is a group in Japan. I find this quite incredible. You can see how small the neonatal hearts are. They're just a couple of millimeters in width, really. So you can imagine the aortic opening is just a fraction of that size, and they've managed to cannulate those tiny aorta onto a Langendorff machine. Of course, this is very, very difficult to do, and this hasn't been widely adopted. I've just put here a table showing, amongst what I've been talking about, some of the different protocols that have been put out in the literature. You can see the different approaches people have used. Some have actually stated their yields. So for example, these two protocols roughly yield about 500,000 myocytes from one neonatal mouse heart, which is not bad. Viabilities are not always great. And interestingly, quite a few protocols don't state one or either of these. So I'm always a little bit suspicious when that's the case. That's the difficulty of isolating the cells. Now, once you have the cells, I'll just talk about the difficulties you might have in actually getting these cells to respond in a classic way. So we know, as we mentioned before, that rat neonatal cardiomyocytes are classically responsive to adrenergic stimulation, things like norepinephrine. So these cells will increase in cell size and area in this case. They'll increase their protein production. And importantly, you'll get typical hypertrophic gene signature changes. So genes like MPPA, MPPB, MYH7, you'll tend to see these things going up. Whereas SERCA2, so ATP2A2 gene, you'll tend to see this going down after adrenergic stimulation. And importantly, these are all things which replicate the in vivo hypertrophic state. So that's why these are a good model of hypertrophy. When it comes to mice, you'll see, and I've just put up one of the real kind of pioneering papers for this, which again was the Paul Simpson group back in 2000, where they directly compared rat and mouse neonatal myocytes and what happens in mouse neonatal myocytes culture. They don't seem to have any response to the adrenergic stimulation at all in terms of things like NPPA, that's the NANF gene expression doesn't really change. MYH7, this is the beta myosin heavy chain, and that actually sometimes seems to even go down, which of course is not what you would expect. And another issue with the mouse neonatal myocytes is in culture, even if you don't stimulate them, they seem to have an autonomous hypertrophy intrinsic to their character. So they will undergo spontaneous hypertrophy, increases in size, and it increases in protein production as time passes. So of course that actually sets a difficult baseline by which you have to try and increase the stimulation of these cells with your stimulation of interest. So yeah, so that's really where the field is, and so the question we have then is, can we modify our protocol, our injection protocol, down to fit these neonatal myocyte hearts? And by doing that, can we isolate neonatal myocytes, and what kind of yields will we get? And will these perhaps be more responsive? So that's exactly what we did. So we've now optimized these protocols. We've just a few things we've done here. For example, we reduced the needle size that we use because these hearts are so small. You've got a picture of the hearts down here. You can see how small they are. That's the atria at the top and then the ventricles below. We use a single flush step. We're using this ETA buffer, so that's just 3 mils into actually both the left and the right ventricles, and that's done within the chest cavity. And then after that, we'll remove the heart. We'll just use 6 mils of collagenase buffer, which is roughly about 5-minute digestion time. And then we actually, rather than using pre-plating, which many groups use, we've optimized a Percol purification step, which we actually reduced now down to about 7 minutes, and so you need no pre-plating. And of course we've optimized the culture media and conditions as well. I've just put here, you can see this is the kind of Percol gradient that I set up, and it's in a smaller tube, so you can do this a lot more quickly. And this is on the left before running the separation, and after running the separation, you actually get the myocytes down at the bottom and the non-myocytes in the top layer. And this is really quite a good separation. These myocytes are really quite pure. So so we've really reduced, so I think it only takes about 15 minutes from sacrifice of the mouse to actually having a cell suspension. And I think what that might lead to, and I hope that you might agree, is that the yield of these cells is really quite good. So we have over 1 million cells per heart, and with high viability. So again, over 90% viability is pretty much what we see almost every time when we do this isolation. I've just put data from 14 mice here, but we've done this with many more now. So the freshly isolated neonatal mouse myocytes, they look like this, so they're mostly round. In this case, that doesn't mean they're dead, they are just round, but they're alive, they're very much alive. But you see a few rod-shaped cells. And after 24 hours in culture, if you see them at the right density, they will form a monolayer, and that monolayer will be contracting in synchrony, as you would hope. I've just put here a picture so you can see the myocytes. This is stained with alpha actinin, so this is myocyte marker in green. You can see the striations, so these are contractile cells, and they've formed intercellular linkages. So you can see that by the connexin staining in red. So this is a monoculture. This is just myocyte. So this is just the myocyte fraction. You see there doesn't seem to be any non-myocytes contaminating in this picture at all. But of course we can, if we want to, we can then reintroduce the non-myocyte fraction into this culture, and we'll have a co-culture model then. So here you can see the myocytes in green again, and the fibroblasts in this case stained with vimentin in red in this co-culture model. Okay, so the important question now, so we have neonatal mouse cardiomyocytes in culture. Do they respond to hypertrophic stimulation? And the short answer is yes, they do. So we can culture these cells in a density that we choose. So for imaging, I tend to use a slightly lower density. For these other experiments, I'll tend to use a confluent monolayer. And these cells have been treated for 3 days in the presence of norepinephrine at 10 micromolar, which is a pretty standard dose. And it's really quite clear to see the increases in cell size that you'll obtain when you do this. And not just that, but this is what was never seen before, but what we seem to be seeing very reproducibly now in our cultures is when you add the adrenergic stimulation to the mouse neonatal myocytes, you'll see an increase in the pathological hypertrophy-related stress gene responses. So NPPA, NPPB, NYH7, like we talked about before, ACTA1, that's the skeletal isoform of actin. These all go up, as you would expect in hypertrophy. And SERCA2, which is ATP2A2 here, that goes down. Also, glucose metabolism goes up. So this is the glucose transporter and an enzyme, limiting enzyme in the glucose metabolism pathway, they will go up. So all of these things are exactly what you would expect to see in the in vivo hypertrophy model, or even in a human hypertrophy patient. So again, this is hopefully going to convince you guys that we are seeing now hypertrophic responses in a neonatal mouse cardiomyocyte model. This is just norepinephrine. I've just put this data here on the slide as well, with phenylephrine, PE here. You see the same kind of responses, sometimes even more actually. And isoproterenol, isoprenolone, whichever you prefer to call it. You do see some changes, but the stress gene responses seem to be a bit less so far in those cases, but certainly they're still there as well. Just to say that the stimulants again were all treated at 10 micromolar for 3 days and refreshed those daily. An interesting thing I thought I would just put up here as well. We had this idea that actually a lot of the pacemaker type cells in the heart are more atrial than ventricular. So it's a question we had. So rather than just taking the ventricular myocytes, what if we combine those back with the atrial myocytes and culture those together? Would we see increased contractility and even responsiveness of those cells? And interesting, that's exactly what we see. So we see increase, the baseline NPPA is higher, as you would expect with containing atrial cells, and the responsiveness of these cells to adrenergic stimulation then in culture is even higher than just using ventricular myocytes alone. All right, so I think that brings to where I will draw a conclusion here. So I'll just say that yes, neonatal mouse myocytes have, I think, previously been disregarded for in vitro models. As we mentioned, because of difficulties with isolation and culture and therefore hypertrophic responses. What I'd like to suggest is that these impaired responses are not something that's innate. in mouse neonatal myocytes. It's probably more a problem that we haven't previously had the right isolation and culture conditions to see the changes which would normally exist. So when we do try and modify our protocol and we optimize this as necessary, as we've shown, and we see high yields, and we see responsive neonatal mouse myocytes in this model of cardiac hypertrophy. And what I hope is that this data now opens the field to researchers or you guys around the world who you have amazing genetic models in your mice already that you can now take in vitro and study the molecular mechanisms of these in more detail than before, using the models that you already have. So that's pretty much it from me. This is our lab. This is Roger Fu is my mentor and PI. So this is actually in Thailand, not Singapore, but it's close enough. And this is our lab having fun on World Rare Genetic Diseases Day a couple of years back. Peter is an animal technician who's really helped me a lot all along the way with this project as well. So I owe a lot of thanks to him. I think he's just here. Okay, so I'm going to wind that up. Oh, just to say, yes, so thank you as well to these guys down at the bottom for having me and sponsoring this work. Yeah, that'll do for me, I think. So over to you guys.
Okay, thank you both Davor and Matt, and this was really fantastic. And I see that we have tons of questions. They're already
stop sharing your screen, maybe just.
So I believe that some of the questions you have already answered. Both of you have already answered during your talk because they were popping along as you were talking. So maybe I would just say that maybe we cut it off there with the last question that was from Helen Collins, and you can try and answer the questions that are already here. Sorry?
Can you stop sharing your screen, Matt?
Oh, yes, sorry, yes.
Sorry.
Busy looking at our Thailand holiday. Great.
So I would say to cut it off at the last question at the moment from, okay, there is another one, Christopher Solis. And because there are really a lot of them, and I guess you will not be able to go through all of them. So please stop. You can start and feel free to stop at any time.
We'll do as much as we can, right? And,
yeah. Hopefully not all of them are just stating that our method doesn't work.
yeah. Well, yeah. I'm sure it won't. Our email, I put my email address up, and I think you did as well, Davor, right? So if we don't get around to answering, then really very happy to answer emails that way as well.
Yes, and actually, you know, the webinar has been recorded or is being recorded, so we will put that up on YouTube channel as per usual. So hopefully that will help as well.
I just got a note from Roger as well on the bottom. He's asking if the attendees can see the other questions. Is that possible or not? I don't really know.
I think they do. I think all the attendees can see all the
I believe they can see the Q&A, right?
Yes.
Okay. There's a Q&A and a chat.
Yeah,
yes.
But let's do Q&A. So start answering questions, otherwise we're going to...
Oh, okay, so that's on us. So I'll go on to the Q&A, right?
Yes.
So
I think what I will say is that I can see some questions here that are answered in the paper. So example, there's one that says why pH 7.8.
So this is from Rodolfo Fischmeister. Hi,
Yes. Hi. So yeah, I mean, we optimized, we tested a range of pHs, right? And this was just the one which seemed to give us the highest yield. We had some speculations about why that might be, right? And I think these are written in the paper as well. For example, it might neutralize acidity, and it might even stimulate some of the enzymes involved in metabolism. So yeah, I think can you see any questions that we have completely never answered, Davor?
Well, I think Matt, I mean, if we just go through all of them one by one, and even if we've answered, if we just give a brief response, it would be nice, I think.
Sure.
So Rosanna from Brazil says, have you tried this approach to isolate cardiac myocytes? So we haven't in Birmingham. I don't know if you guys have in Singapore.
We have. Yeah, it works. Yeah, yeah. Of course, it depends a little bit on the size of your rat. If they are pretty young rats, they're not always, you know, that much bigger than a big mouse. So it's pretty much the same then. I can't remember off the top of my head how many grams that would be. But I know that once you start getting to big rats, then it becomes a bit more difficult, because you really have to be pumping through so much enzyme that you can do it. It just needs a lot of enzyme. So I think basically you try and scale what we have to the mouse, but increasing relative to the size of the rat, right?
Sorry, go ahead.
I was just going to say that there are actually a couple of papers who have detailed some modifications of our protocol that they've used in rats as well. So again, off the top of my head, I can't remember those, but I could send them to you if you want to distribute them to people.
Yeah, and I would probably say that most people don't struggle isolating rat cardiac myocytes. It's the mouse cardiac myocytes that are trickier for most labs. So
they are tricky, yeah. But I guess if your lab doesn't have the Langendorff setup, you'd still need to have something.
Okay.
Should we go to the next one?
Yeah, you go ahead.
How many days can you keep? How many days can we keep these cells in culture? Actually, a lot, but they won't be staying the way they were. Right. So the longer you keep them in culture, they will. I mean, you showed Davor how the cells will de-differentiate and redifferentiate. That cycle will happen a lot in the first week, especially if you add serum during that time. I think if you want to do long-term culture, that's where, I mean, really long-term culture, like in the kind of weeks type level, then that's where I would usually add some serum into the media to keep them viable. Maybe 1% would keep them viable more. And we sometimes would use the lipid concentrate as well, which they seem to like. But over that time in culture, they will be quite different to how they were when they were in the original mouse heart. Right? So I think if you want to do that, it really depends on what you want to use the cells for. Neonatal cells are a bit different, because when you can keep those in culture, we've done that for about a month so far. And actually they seem to mature towards a more adult phenotype in that stage. You just have to keep making sure you change the media every other day. And then they really develop some quite nice striations, and they become elongated. So they at least look more like adult cells after that month. I don't know on a kind of electrical and physiological level how adult they are, but they look like it.
Okay. So Celia Fernandez-Sanz says, what about stability in time? Are there any differences? Do they last longer? What about T-tubule stability? I mean, we've not compared, have we, between the Langendorff and the non-Langendorff in terms of what happens to T-tubular structures?
No, I don't think we have.
So, but I mean, I published a paper in 2011, I think, in Journal of Experimental Physiology, looking at differences in detubulation between mouse and rat cardiac myocytes. And obviously they do detubulate when they are in culture, but I've never, in my earlier work, never took them, longer than 4 days. But obviously what we're seeing here are changes in sarcomere structures beyond that time, in the presence of 10% FBS, right? So
yeah, they definitely change. If that question is asking about the difference between Langendorff and injection protocols for that, then I don't know. I don't think there's anything to suggest that they would be different. But we can't, we can't be sure is the answer to that
nothing
question.
from the basal functional and signaling data that we have. You want to take the next from Adam Vittetto? Because I think Danny is just kind of...
Yeah, he's answering, doing our job.
Thank you.
For those investigating left ventricular stretch activated channels, do you think latent SAC potentiation could result from direct perfusion of the left ventricle. I think this one may be for you, Davor.
So, put another way, would non-Langendorff isolated left ventricular cardiomyocytes be a good model for investigating stretch-activated channels? I mean, I would argue that you're going to get stretch activation. Well, actually, no, I see the point. I don't know.
Probably get more stretch. It's probably you'll get more stretching in our injection method.
You will, but then again, the calcium levels will be lower because of ETA. So I don't know. Adam, I'm not sure. We haven't done it, so I can't comment.
Any idea whether micropattern cover slips would help? We haven't looked at this. Have you looked at this?
So this is a question from Giulio Agnetti, asking whether micropattern cover slips of dishes would help maintain morphology. Obviously a lot of people are using these for IPS differentiation. I'm not familiar that anybody's used them, and maybe there are people that have used them for adult ventricular cardiac myocytes, potentially, because they do help with IPS derived cardiac myocytes to differentiate into more adult-like phenotypes. So maybe they do actually preserve adult morphology as well in the adult ventricular cardiac myocytes, considering we've seen the plasticity going both ways, right? So to me, it would make sense. We've never tried it, but I think, Giulio, that's definitely a good experiment for you to try out, because it may help preserve long-term culture conditions for adult.
It would depend on the kind of patterning as well, right? One of the things about adult myocytes is they're very rigid. So when they fall down onto the tissue culture surface, they don't really move around or migrate. They just stick where they are. If the patterning is in the right direction for the direction that they've landed in, I could see that might well help. However, if the patterning was in the opposite direction, would it make it worse? I'm not sure. They might. So I don't know. It's a really interesting point. Rodolphe, next one, shall we?
No, go ahead, sorry.
No, I was just going to say, should we go to the next one? So Rodolphe is asking the pH in the solution injected in the apex.
So he's referring to his previous question about the pH 7.8. Why do we use it? And
right.
So I think you've answered that, though.
I think we have addressed that, so maybe you can move on to the next question.
Have you tried culturing? Okay, so we just answered this one. That was the same idea that I had as well, and it's interesting. Assuming that you mean neonatal, I don't think
they do, because it's been up at 5:16 p.m. So whilst probably
okay. Yeah.
So we have in Birmingham, we haven't.
I haven't done this. I'm not sure with adult if it would make that much difference, because you don't generally form a monolayer with adult cells, right? So you might have atrial cells there, but I'm not sure they would really be touching and communicating electrically with the ventricular myocytes, unless you're thinking they might release some factors into the media which would have an effect, maybe.
Paracrine factors might play a role.
Yeah.
We know that works with cardiac fibroblasts and cardiac myocytes. So maybe.
Right. Yeah.
But we haven't tried it.
We're fumbling around. Okay. Why do we think? Should I read this one? Why do we think mechanistically I think that's cardiomyocytes retain their morphology? Yeah, it's a good question. In pH 6 versus pH 4 when cultured. Good question. I don't know. I don't know the answer to the question. Can you think of anything, Davor? Why the?
No, I can't. I think KC. This is KC from Oxford. I think what's peculiar about this is that actually some of them, the ones that are rectangular, effectively retaining morphology, are not necessarily alive. So, that's another sticking point in this. But
that is true.
I don't know.
I think at least for the first few days they are alive, and the more acidic pH, it does reduce their remodeling. Whether it's just making them less healthy, I don't know. I don't know if that's why. Yeah, sorry, I don't know the answer to that question.
I think this next question is definitely for you.
Why not start the culture with FBS to try and prevent the dedifferentiation? Okay, so FBS actually drives dedifferentiation as well. So it drives a dedifferentiation, redifferentiation cycle. So we do sometimes do this. You can start the culture with FBS. Within just really a couple of days, you'll see a rapid dedifferentiation, rounding of the myocytes, followed by reforming of those myocytes like Davor showed. Pretty much like they looked after 2 weeks in those pictures you showed, Davor, they can start to look like that after just a few days if you start the culture with FBS already there.
Yeah.
Yeah.
So, I mean, I can get, so Roman Perrier says, maybe I missed it, but which collagenase did you use? So we used a combination of 2 and 4. But actually, we have also used other combinations as well. And as long as you don't overdigest, obviously, as long as you get it right, the right activity, it didn't seem to make a huge difference. But I think you still have to, just like with the Langendorff method, you've still got to test out your collagenase activity and find the right one. That was at least the case with us.
Yeah, I agree with that. It's you can probably use many different types. You would just have to test it and find the right concentration. But if you're using 2 and 4 like we do, then usually roughly the concentrations that we suggest, they usually work. Yeah.
And adding a protease inhibitor as well helps. So
a protease. Did you say inhibitor?
Inhibitor, yes.
Do you add a protease inhibitor? Which one?
I can't remember now. I can tell you.
Because we add a protease, so we're doing the opposite. We use protease 14 alongside the collagenase.
Okay. Well, we've copied obviously all your work initially, but
some people do. Yeah, they use soybean trypsin inhibitor, but that'll be later on. Yeah.
Okay.
Next question. Does co-culture with fibroblasts affect how long you can maintain the viability of myocytes? I don't think I've ever directly looked at that. No, I can't answer that question. Really sorry. I wouldn't be surprised if it did, but I don't know, and I don't know in which direction it would either, actually.
I think the problem generally with that is that obviously cardiac fibroblasts will keep proliferating, so until they reach full confluency. So
yeah, I mean, I've had situations like that where we've had myocytes and the fibroblasts just kind of grow around them, and you get a monolayer. And the myocytes, they look very happy to me. Okay. They're still beating, they're still beating away. So I haven't seen anything myself to suggest it would affect their viability. It probably would affect their physiology. Anyway, we can try it. Next question, yesterday. 12:22 a.m. So that would be the adult myocytes, do you think? Adult myocytes? Yeah, I think you need laminin usually.
Yeah. So this is from Delphine Mika in France. Hello, Delphine.
The neonatal, we just use 0.1% gelatin.
Have you tried, if I may, in that sense, have you tried other substrates for plating the adult cardiac myocytes to see whether that might affect viability and how long they last? For instance, matrigel or something like that?
We haven't. So
yeah, I've tried a few. I've tried quite a few, and to be honest, their adherence to anything other than laminin is pretty poor generally. There is some, I think there was some adherence to collagen 4, but again, it wasn't anywhere near as strong as laminin, and I don't think I followed up on those experiments, so I couldn't say past that really. But generally, I think laminin, it's the best bet. I've tried on major gel. They didn't seem to enjoy being on major gel as much. What's that other? It's not major gel. The equivalent of major gel from Thermo Fisher. So it's anyway. Yeah, it's kind of that thing, but I think that worked a bit better actually. But again, laminin itself was always the best in my hands.
So, Rehan Babai, very good talk, thank you. Did you add FBS after day 8 to regaining sarcomeric structures or in a separate experiment from day 0? I thought it was added afterwards, right? Is that right?
Yeah, yeah. In that experiment, that was added afterwards. Yeah, yeah. We've done it. Adding, as we mentioned before, we've done it earlier as well. You just get that phenotype quicker, basically. Next question: How long are the cells viable? The major concern is cell viability, which falls rapidly. Yeah. Whether preconditioning the cells in serum will help in better cell viability. So I think we've, I could address this attendee to the paper where we've done quite a few studies on viability in culture in many different conditions, right? Yeah, the viability really drops off quite fast in the first few days, but after that, the ones that have stuck down, they tend to stay stuck down, and then generally, especially if they start to remodel, actually that seems to be a bit protective. So even though you might not want your myocytes to remodel too much, but if they do remodel, they probably survive a bit better in culture. Preconditioning in serum, I'm not quite, do you know what that might mean, Davor?
I think that they literally just mean, so I think Vibarani means probably just whether sticking them in serum from day 0 would help the viability. And of course, I think it would, but it would also cause differentiation from, from day 0. So
yeah. There's one thing that, so previously I've mostly used 0.1% BSA in the media. Sometimes I swap that out now for 1% FBS. I think 1% FBS, it does improve their viability, and it doesn't seem to cause too much dedifferentiation as well. So if that's an issue, then that's certainly something that you can try. So
I'll answer Michelle Russo's question because I think I'm perfect for that. So Matt is an expert in the technique and very good. I'm really not. So actually I can answer this question. How important is the operator handling in the non-Langendorff methods? So for example, is the pressure of the pump that is more consistent better than the hand method? So I would say, because I am terrible at this and I tend to vary the pressure a lot, I definitely find that the operator is often a source of error. And I do think that maybe it relates to some of the stretch channels that were referred to previously. So I actually know this even because when I sort of inject the hearts with the collagenase, quite often the atria blow up. Whereas when Matt does it, atria stay pretty flat, okay? Because, and presumably I must probably just sometimes rupture the valve or something like that as well. So I think there is a operator error that can occur and that can induce changes in viability. That's for sure. I don't know what you think about this, Matt, but.
Yeah, I think that generally this is one of these techniques that if you do it quite a bit, then you will quite quickly get the hang of it. But if you only do it very sporadically, then it just becomes, it becomes, your results become sporadic. Personally, I much prefer to do this in a manual way than using an automated pump. I think that you have so much more control, and each heart is a bit different. So it's very hard to set that on an automated pump exactly what you're going to need and the direction you want to push in and things. Whereas manually, you can do that. But yeah, I think it's a good point. Maybe if there's people who are not doing this very much, then something in a more automated way might be beneficial.
So, I mean, actually, I'll answer the next one because it's referring to me. So Claudia Sacchetta is saying that they're using this method and they observe pretty low viability after digestion. They try reducing the time of the digestion, concentration of the enzyme, but are still having problems with the optimization. Which part of the technique do you think is crucial to obtain a high number of viable cells? So I think probably our previous response to the question answers this to some extent. Perhaps I think the operator is a part of this, but I don't know, Matt, maybe you can contribute to this as well beyond what I just said.
So what I would say in this situation is drop us an email, because we've had a lot of people emailing about these things, and we can usually work it out. It usually requires a few questions to find out exactly what's going on. So from this, I can't exactly tell what's the situation. I know, for example, that she suggested that she's reduced the digestion time and the enzyme concentration. So it sounds like she's suggesting she thinks it's an over-digestion. I would be surprised if that's the case, because it's not usually the case. If anything, it's usually an under-digestion is the main problem. And it often comes down to how quickly and efficiently people actually start flushing the heart with EDTA buffer after sacrifice of the mouse. So if you don't do that quickly, then you might get blood clots inside. That seems to be quite a frequent cause of poor harvest yields. But yeah, what I would say is drop us an email. We can probably troubleshoot that.
I mean, Matt, a question about that is how long does it take you to inject the EDTA into the right ventricle when the heart is still in the chest? And once you've obviously from the time you open the chest to the moment that you've actually started to inject EDTA.
Okay, yeah, so we do that pretty fast. And what I would say is that this is quite dependent as well on the method of euthanasia that people use. So generally, if people are using isoflurane like we do, you can really shorten that time, because you're almost doing a surgical operation then. So you can be right there ready with the chest open at the point of basically animal sacrifice, right? However, when people use things like cervical dislocation, then that mouse might then be sacrificed, a minute before the chest is even open and the heart is reached. And in that time, you inevitably get blood clotting things. So we do it pretty quickly. But it doesn't necessarily have to be the case. I think that what I would usually recommend to people is that if they are inexperienced in this, or they don't have access to an isoflurane system, then try with heparin pre-injection first, because that will just slow the rate of, the blood shouldn't clot then, right? And so you then don't have that same problem. And that might solve the issues. And then, of course, once people get more experience, they can probably not need to do that anymore. Yeah, that would be my suggestion.
So,
okay, Rosanna Bassani, how do you retrieve isolated cells as to stop the enzymatic digestion? So this is really a case. So once the tissue has been, the enzyme step is complete, we then tease the pieces of tissue apart and triturate with the pipette. Then we just add stop buffer. So that stop buffer contains FBS, and that FBS inhibits the collagenase enzyme reactions. So that's what stops the enzyme digestion then. Yeah.
Okay, the next question is really long by Charles Chung.
Oh, wow. Yeah, BTS is something that I think is a contraction inhibitor as well.
Yeah.
Okay, so what's this question getting at?
So basically, it's talking about the fact that all these contractile uncouplers help you maintain cells in culture.
Right.
It's asking about the mechanism about that through which this happens.
Mm-hmm. So yeah, so this might be, yeah, I think it's probably a good point that some of this is due to reducing ATP depletion in the cells. And the question asks, I guess, so is one mechanism by which the low pH preserves the cells does that happen through inhibition of SERCA or myosin ATPase? Maybe if they're, I don't know, are those inhibited by low pH, to your knowledge, Davor? Maybe.
I mean, possibly. I don't know. I really don't know the answer, I think, without testing, but it's a good hypothesis. So that could be the mechanism.
So which inhibitors of contraction are you using in your culture?
So in culture now, actually, I prefer to use blebistatin. I think it's a bit cleaner than BDM. It's a bit too expensive to use blebistatin for the actual digestion step itself. But once we get the cells into culture, that's what I would choose. And your concentration of that can be pretty low. Between 1 and 5 micromolar is usually more than enough. I wouldn't recommend going above 10 micromolar. Some protocols say 25, but I think I saw reduced viability at 25 micromolar. So yeah, 1 to 5 micromolar blebistatin, I think that's pretty good.
Yeah. And it will still, I mean, even blebistatin at those concentrations will still affect your electrophysiological properties, but not to the same extent as BDM. You know, for example, we use it in optical mapping a lot, but we don't use BDM, for example.
Right.
The effects are quite severe on electrophysiology as well. So, okay, so
I guess just the last point of that question: what is high EDTA flush valuable in increasing cell viability by reducing calcium overload? Yeah, I think that's definitely true. Yeah. So in answer to that question, yes.
Yeah, but you know, but there it is a trade-off, because I think if you reduce that calcium severely, then you've effectively got cells with very low calcium. So if you're doing functional experiments like calcium transients, contractility, then actually that can be quite difficult. So these things need to be considered actually, and you know, I know that some labs, and including us, are kind of reconsidering whether to go with such a high EDTA if it's possible to titrate it lower. So it, you know, this methodology is not, I would say, 100% proof at this stage. I think it depends on what you need to do. You need to consider what you're adding, as with any methodology.
Yeah, and what you want to use the cells for as well. So I guess, you know, you could probably get away with using much lower EDTA if you wanted to. You might not get as many cells, but if those cells are better for what you want them for, like electrophysiology, then, you know, maybe that's a better approach for you.
Absolutely. Right, should we move to the next one, do you think?
Do you want to go for this one, Davor?
Dan Johnson. Hi, Dan. So, hadn't seen the data on the re-establishment of sarcomeric structure after sustained culture and potential spontaneous activity. Well, we don't really report any spontaneous activity. Oh, you mean beating? Sorry, yes.
They do beat, yeah.
They do. Have you done any electrophysiology in these cells, and have you checked for expression of HCM channels, for example? And so we haven't. Well, we haven't. I don't know if you guys have.
No, we haven't.
Yeah, I mean, Dan, I would say, you know, this is the reason why I presented that is because it's a peculiarity that tells us maybe something about the cellular physiology and plasticity, but I wouldn't necessarily, and maybe for developmental biology, this would be an interesting thing, but, you know, for us electrophysiologists or cellular biologists that kind of care about pathology, I I wouldn't spend a lot of time on that, but that's the reason why we haven't pursued it.
So guys, do you maybe want to pick another few questions to address now? I leave it up to you, obviously, to decide.
I mean, I'm happy to keep going if you are.
So I mean, I think these webinars open a possibility to crack a bottle of wine.
Yeah, we should
a bottle of wine and get some chocolates or whatever you fancy. And as long as the people that are within the webinar, including yourself, Nina, by the way.
Yeah, I'm happy to stay. I'm just saying, if you're tired, you have already answered many questions, and there is a total of 56 questions. So
shall we see how far we get
through?
Yeah. I mean, the number of participants is still very high. So if you're happy to keep going, I think that people will be happy as well. So
sure.
I'll leave it to you because, Matt, for you, it's 1 o'clock in the morning or whatever it is. So whereas for us, it's normal time. So
I'm on UK time anyway. Anyone in my lab will tell you that. So.
Okay, fine. Okay, so then we move on. Do you want to take? Because this is neonatal.
Yeah. So I just I'm not sure if this is a question or a exclamation from Rosanna again. The intraventricular perfusion technique works well for neonatal rat hearts. Is that something that that they've tried? Like, it's great if so. Then
yeah, I think it looks like to me like they've tried it.
Okay, great.
So, but that's what I understand from that question. Well, from that statement.
Excellent. Okay, well, I mean, drop us an email if you have data on that. It would be interesting to see it.
Definitely. I mean, I'll take next one because obviously we've done a lot of the calcium transient stuff. So from Purvi Trivedi, did you compare the cardiomyocyte contractile properties and calcium transients between day 1, day 3? Post-isolation, the answer is no, we haven't. So we only study these properties on day 0, effectively, after isolation.
How easy would it be, Davor, if you wanted to do that? So if you wanted to look at calcium transients and electrophysiology after some days in culture, is that easy enough to do?
No, I mean, I've done it up to 2 to 3 days.
Okay.
But often the contractility goes down. So, you do lose as the cells start to sort of de-differentiate, lose sarcomeric structures, lose T-tubules. You obviously start losing the sarcomeric force as well. You start sort of altering the passage of calcium channels through the cardiomyocyte. So these things will become more difficult. It's also more difficult often to patch clamp a cell, which has got a bit more of a blebbier membrane. So.
Okay, so this is another question addressing low yield or variable yield when using the injection protocol. And that can vary even within the same batch from one mouse to the next. Was this due to the operator or something else? Again, these things, I think, Robin, drop us an email. We can discuss and try and troubleshoot. That sometimes there's some very small things that come up. We can be having a conversation with someone who's trying to troubleshoot their protocol, and about 3 emails in, it comes out like, oh wow, oh no, do it this way, and then it's just problem solved. So it could be something like that. It's very hard to say here. So I think we'll leave. I think what we'll do, if we come up with questions about people who have suffered low yields, then shall we just skip those for now and say, drop us an email, and then we'll certainly address that, right? Let's do that. Emma Robinson, hi Emma. So she's been using our protocol. Hints and tips for subtle differences in method depending on downstream applications. That's a good question. Do you want to take that one, Davor?
So Emma's saying that she's been using the method since 2017 herself. In her experience, preps that yield good myocytes for electrophysiology are not as good as those that yield good myocytes for long-term culture. Any hints or tips or any differences? Is there a digestion time? So I personally think that if you want better quality cells for electrophys, generally speaking, whether you use a Langendorff method or an injection method, you want to underdigest and accept the fact that you will lose a lot of the cells, but at least you won't strip off the proteins on the membranes through the action of an enzyme. And therefore you'll get a smaller yield, but they're more likely to be amenable to patch clamp because you can achieve better seals. And if you want cells to, for high yields, such as, for example, I don't know, adenoviral transfections or long-term culture, then you sort of digest more and then you get more cells, but obviously they might not be as amenable to patch clamp, for example. So that's what we tended to do with both Langendorff and injection methods. So that would be my advice. Now, I don't know if you can comment on that, Matt, but that's often what we do, generally speaking, regardless of the methodology.
Yeah, I think I completely agree. Yeah.
So Emma, hopefully that answers your question.
Okay, this question, again, it's from Rosanna about avoiding do you think they mean EDTA?
Yeah, so thanks. Well, they might have used EGTA, actually.
Yeah. EGTA you could also use. Is there a way of avoiding it having access to this? So again, I think it's this question of the calcium chelation and having a deleterious effect in presumably some functional study.
Yes.
Yeah, I guess it comes back to what you were saying, right, Davor?
I think it depends on what you want, I think if you drop this calcium all the way, yes, you get cells that are more viable, but also functionally they're a bit less useful. So it depends on what you want. This is exactly why I said it earlier that we're definitely thinking about the extent of calcium chelation. That definitely needs to be modified depending on what your usage is. But you've got to consider the consequences of that as well.
All right, I can take the next one. So this is Sina. Given the immature state, have we observed any cell division of the neonatal myocytes? Yeah. So so we use neonatal myocytes from pretty much within the first 24 hours after birth. At this stage, some cells are still proliferative. I'm not sure if we've done this or one of our collaborators did it. We did BRDU staining or EDU staining, and you certainly see some nuclei lighting up for that. You also will see some binucleation. So I guess when you see EDU staining, for example, it's not necessarily a cell division. It could be a binucleation or something. I do think that we see some neonatal cells dividing, and those two daughter cells, their contractility is not always good. So, for example, when I'm doing studies for the hypertrophy model, I try and keep the cultures relatively confluent because I think that reduces the proliferation of the cells, and then you'll get better contractility across the monolayer by doing that. Okay, Lisa has asked, are we able to isolate microvascular endothelial cells from the adult or neonatal preps using a gradient or different culture media? It's something we're actually very interested to do, and I think we will do it. It's the endothelial cells are definitely there. I can say that for pretty sure. However, to isolate them, I don't think that you would get a pure population of those cells just by doing a gradient, because the density of those cells will be very similar to other non-myocytes. So there's kits from companies like Miltenyi for specifically isolating endothelial cells, I think that's probably the way that we will go. And just, yeah, so we'll aim to get pure endothelial populations that way. Okay.
So the next question, I think you have already addressed, right?
Yeah. Yeah.
Concerning the different substrates. And then there was a question from Robin Colpert that has been kindly addressed by Emma Robinson in the chat. So
okay, great.
if you want to add
Thank you, Emma.
please feel free. But I think also that has been largely addressed. So if you want to move on to...
Oh, okay. I think that's one that we spoke about already as well, actually.
Yes. And Robin's actually texted all of us saying thank you for answering the question. So
yeah. Oh, that's very nice. Thank you for listening. Roycin Kelly, does the method of euthanasia of the animal, and in terms of NC3Rs, is it possible to use hearts harvested from animals that have been culled for other purposes? Yeah, interesting question. So this comes back to something we said before, I think, is that really you want to try and get to that heart as quickly as you can, otherwise blood will start to clot. And once blood clots, you're not going to get good perfusion of the coronary circulation. So I wouldn't recommend this, but if you do, then you might be able to get some myocytes if you pre-inject that animal with heparin, is what I would say.
Yeah, and isolate the heart first. So, you know, and then
yeah,
you can deal
ideally, with ideally, I mean, the question says if it's not the first organ harvest. Oh, yeah, okay. So if possible, yeah, do it as quickly as you can.
Yeah. So I would say, Rosanna, I mean, I think it's an important NC3R question. So for those of you who don't understand what this means, it's National Institute for Reduction, Refinement, and Replacement of Animal Research. And we try and optimize our experiments to actually make full use of the animal being sacrificed, which is important scientifically, but also ethically. And so in our lab, this is what we do. We collect other tissue as well, but we collect the heart first, and then subsequently the other organs. So it's an important question to ask.
And on the method of euthanasia as well, I would always say, if possible, use isoflurane with oxygen. That is just the most likely to give you the best yield in my experience.
Yeah, I think this one's yours because we don't use Percol. So from Charles Chung.
Yeah, okay. Hey, Charles. Excellent work. Thank you very much. Would the Percol step used for neonates also help in adult myocyte isolation purification? I have done it and it does work. It's just, I don't think it's necessary for the adult cells. They fall down by gravity quite quickly, and it's a good way of, you can do that at the same time as you bring up their calcium concentration as well. So it doesn't really save you much time to do the Percol. And I always think that if you can avoid spinning the adult myocytes at a high speed, then it's best to avoid that. So it does work. And I guess the times you could use it is if you don't really care if the cells are that alive afterwards. So say, for example, you just want to get the myocytes to isolate RNA or protein from them. Yeah, that might well work. You have to do some adjustment of the actual density of the Percol that you use. But it's certainly possible. It can be done.
So do you mind if I say the next one?
Yeah.
Because I've read it. So
yeah.
So Ken Ginsberg saying, thank you for this methodology. There are 3 things that they consider in their labs, and I fully agree with everything they say. We've already covered it actually, but I think it's important to emphasize. So number 1, level of anesthesia, controlled dosing using isoflurane is important. Number 2, calcium reduced to micromolar rather than nanomolar levels, which again, we've discussed quite a lot. And choice of enzyme would balance the pure collagenase versus non-specific protease activities. So empirical trial and error. So these 3 things govern successful isolation, be it injection or Langendorff. I fully agree with that, Ken. And I think it's important just to emphasize that again.
That's actually the reason why we use collagenase 4 and 2 mix. Did you get that? I actually want, because we got the recipes for these enzyme buffers from previous publications, and they tend to use collagenase 2 and 4. And in some ways, I think, why not use one or the other? Apparently, it's because of the balance of the actual collagenase enzyme activity, but also the non-specific protease activity of that collagenase prep as well. So collagenase 2 is a bit less pure, if I get that right, than collagenase 4. So collagenase 4 is more specific for just cutting collagen, whereas collagenase 2 will cut collagen and a few other things as well. And you need a bit of both of those. Yeah.
I'll trust.
Yeah, well, I mean, don't take that as gospel, but that was my understanding of it.
I think this one's for you again from Chris.
Fantastic. Yeah, great. Thank you, Chris. For the mouse neonatal work,
Chris is my PhD student, by the way, so it'd be nice.
No, I remember. Yeah, it's great. Have we tried culturing the HL cells alone? That's a good question. And actually, no, I haven't. I never have done that, but we should actually. We should do that and see how they behave. Yeah. I just wanted to have it because the number of atrial cells is much lower than the number of ventricle myocytes. So you would probably have to get a lot of atria before you have any kind of confluent monolayer from that. But it would be interesting to do. I just wanted to put some atrial cells in there in the hope that there would be some pacemaker cells, which could actually stimulate contraction in the monolayer. And yeah, I think they did actually. I think they did. We haven't. So this is a question from Helen Collins. What does the calcium transient or handling look like in the? She says neonatal rat ventricular myocytes. Maybe means neonatal mouse ventricular myocytes. But either way, I'm sorry, we haven't actually looked at this yet. But this is something Davor, you suggested we should do actually already. So yeah, there's more independent confirmation that we should do it, right? Yeah, so we should do this is the answer to that question.
Yeah. No, I mean, it's something we discussed actually just today.
Yeah.
So we'll probably, I mean, we're about to submit this, so we'll probably be asked this.
Yeah. So if you're reviewing, please be nice and we'll do it, right?
Shields enough to run Western blots by Chris Solis. I mean, presumably meaning regarding the neonatal isolations.
Oh, plenty. Yeah, plenty. I mean, for some perspective, a neonatal heart has maybe almost all the myocytes by number that the adult heart has. And so if you're thinking of things like DNA, they actually have about the same amount of DNA, at least myocyte DNA, than the adult heart has. Although, of course, if the cells become binuclear in the mouse, then that will change. But of course, in the neonatal heart, you have less cell mass, right? So there's less protein there. However, you still get 1 million cells, and 1 million cells is going to be enough for. Let me get this right. So say if you plate these cells about 100,000 cells per cm squared, so about 10 cm squared, it's going to give you a 6-well, one well of a 6-well plate, right? That's more than enough for protein. So yeah, there's plenty for Western blots. Yeah. Adult rats, we mentioned this. So Tejas has asked about applying this to adult rats. Yeah, it can be. How do you pronounce his name? Javier Laura is being checked the number of events from different cell populations after FACS. We haven't compared between Langendorff and our method for that. We haven't compared.
No, but what I would say is that, I mean, you've done the FACS analysis of non-cardiac myocytes, and the numbers are pretty much what you would expect.
Right, yeah.
Proportions are what you would expect, probably. That's probably a better way to say it.
Yeah. Yeah. I think if you want to do this, then one thing you might want to consider is when you're pumping enzyme solution into the heart, that enzyme solution is picking up cells on its way as it goes out of the heart as well. So if you really want to try and collect all the cells in the heart, you don't want to discard that effluent digestion buff. You need to keep that, because I think a lot of endothelial cells might actually be in there. They'll be the first ones to get flushed out. But it's certainly something that's quite easy to do, I think. Do you want to do this one, Davor, from Patricio?
I mean, it's a fairly simple one, isn't it? Because actually, I'm following what you did and suggested at the start, which is a third, I believe, of a culture medium.
Yeah. So I don't actually measure the calcium specifically. I just ramp up to whatever buffer the cells are going to be in eventually, which is usually physiological calcium. So you can start with 1/4 of your culture media, go up to 1/2, then go up to 3/4. Then after the 4th time, you're in your pure culture media or thyroid buffer or whatever you want to use, right?
Yeah. So I mean, we've had successes with basically starting from 250 and then going essentially, that was 1/3, and then going to essentially 2/3, and then effectively 100% 3/3. But I do think that it's probably better to start lower if you want to retain more cells. And then just have more sequential introductions. So, but I think that the same applies to a Langendorff-based isolation method as well. Okay, so from Emiliano in Brazil. Hi, Emiliano. Any ultrastructural studies? What about T-tubules? I mean, we've not looked at this. We've not looked at T-tubular structures, but functionally, these cells look very healthy. Therefore, I don't expect any T-tubular modifications compared to a standard Langendorff setup, unless he means maybe to your neonatal cardiac myocytes.
I think the question might be at different times in cell culture. So this is in cultured cells. How quickly do the T-tubules, they deteriorate over the first few days in culture, I think. And that's pretty standard with any protocol.
Yeah, I mean, both detubulation as well as the actual organization is affected.
Yeah, we haven't specifically looked at that, but I'm pretty sure it happens.
Yeah.
Oh, look, the next question we answered it about the 2 different collagenases.
Nina will be happy.
So yeah.
Great job.
Sorry, Nina.
Yeah. So, okay. From Barbara, great talk. Thank you very much. What do you think about using this technique on large mammals? Yeah, it's we get asked this sometimes, right? It's a difficult one. I don't think you could do it. You know, for example, if we're talking about, say, a pig model, there's no way that you could have that much enzyme pumping into a pig ventricle to I don't think it's really feasible. Have you ever done it, Davor, where you've actually picked out an artery? No. And yeah, so I've heard people who've done this kind of thing where rather than use a whole heart from a larger animal, you just take a piece of that heart which has some vasculature in it, and you actually cannulate that artery, for example, and you perfuse the tissue through the artery with your dissociation solution by doing that. I haven't done it myself.
No, we've not done it.
There's protocols online for dissociating myocytes from larger animals and also human tissue, which involves almost taking a microtome and cutting very thin sections. So these are about 200 micrometer thick sections, and then digesting those in buffer. And we haven't actually had that working yet, but a number of people have, and they seem to get quite good yields by doing that. So maybe that would be the better way for large animals.
Yeah.
Nicholas has asked, have you been able to culture isolated adult cardiomyocytes with your method? Yeah, again, I would, adult, yes, it says adult mouse card. Again, Nicholas, if you take a look at our paper from Circulation Research in 2016, we address that question in quite some detail there.
And from Andres Medina, I can respond asking if we use BDM in the isolation buffers when measuring calcium transient sarcomeric length. Yes, we have, but as I said before, obviously the BDM is not present in our buffers when we're recording calcium transients or sarcomere length shortening. And actually, the quicker you wash it out or the lower the concentration that you use, the more successful your experiments will be. So that would be my advice.
Mm-hmm. Okay, question from Igor about, this is what we just spoke about, isolating human ventricular myocytes. Yeah, Igor, try and find the papers where they have done this using effectively a microtome.
Igor will know these. He's done a lot of work on this. So.
Oh, okay, then probably you know more than I do.
He does.
but there has been a recent, what was the author? It was in Nature Cell Biology, a single cell paper from human hearts. Again, used a modification of this protocol and made some small changes, which they said gave improved yield. So don't take my word for it, but take theirs. I think they got some really nice data from that paper, a very good paper. So yeah, maybe have a look at that.
So Amina, I think this is your question.
From Amina, interesting talk, the neonatal mouse ventricular myocyte monoculture. Is that how the cells look pre or post stimulation? That was pre-stimulation. Also, you showed qPCR measurements for hypertrophy. Did you do cell size measurements? No, I haven't done quantification of cell size yet. This is something else we said we need to do, right? Davor, you suggested that already.
It will be done.
Yeah, we will do it. Yeah, we will do it. This is just something which I just visually, it's quite striking visually. I'll be very surprised if that doesn't come up as significant when we quantify it, but we will do that.
So I'll take the next one. Maria Fernandez Velasco. Did you try other enzymes besides collagenase, such as librases? So we definitely tried librases, works really well. So in fact, librases worked really well in our hands. But sometimes you run out of libraries, you can't order it. So yeah, but I would definitely encourage use of libraries actually. So.
Okay. All right, I'll take the next one then from Sankar. Thank you very much. I have doubt that hypertrophy is a condition of adult heart to be more specific, completely matured cardiomyocytes. So I think what Sankar is saying is that it's difficult to model hypertrophy with adult myocytes and in vitro, and I completely agree. It's very hard to measure hypertrophy, especially in terms of things like cell growth. People have done it with increased protein production. If you can do that, but you sometimes need radioligands for that. But yeah, this is exactly why we decided to try the neonatal cardiomyocyte approach, which is, I think, much better for hypertrophy modeling than adult myocytes. Yeah. Okay, so this is I'll take this next one as well. It's an anonymous question. Why would the contractility be better when you add atrial cardiomyocytes into the ventricular cardiomyocytes rather than ventricular alone? So the idea when I did this was because myocytes will spontaneously contract on their own. They have an intrinsic contraction, but of course, in your heart, especially as you get older, usually the contractions are controlled by pacemaker centers in your heart, and those are more atrial than they are ventricular. So the idea of this was that if we could include the atrial cells in the cultures as well, then some of those pacemaker cells, which hopefully have high rates of spontaneous contraction, will then be able to send their signals out to the neighboring ventricular myocytes and cause that whole monolayer to contract at the rate of that fastest contraction rate. So actually that paper I showed from 1960, I think that was one of their observations, was that when the neonatal cells form a monolayer, they tend to contract at the frequency of the highest frequency contractors in that monolayer. So that's, I think, what we're trying to get at here. So if the atrial cells might be contracting faster, or at least more predictably, then they can induce the whole monolayer to contract more predictably. So that was the justification for that.
So you want to go for this one? Junhui?
Yeah, Junhui, this is, I think they mean BDM. Why do we include BDM in the digestion buffer? Yeah, so we address this in the paper, but this is a contraction inhibitor. People don't exactly know all the ways it works, but we do think that if you can reduce myocyte contraction, you'll probably reduce ATP depletion in those myocytes. And ATP depletion is one of the major ways that myocytes will die during isolation.
Yeah, I don't really understand. I think we've answered collagenase isolation.
Yeah, okay. I think, yeah, hopefully we've addressed the next question already.
I can't answer this next one.
Okay,
So
yeah. So this is a question about cytosine arabinoside. So this is a proliferation inhibitor, which is sometimes added to usually to neonatal myocyte cultures, particularly neonatal rat myocyte cultures. The reason people add it is to stop fibroblasts from proliferating and taking over the culture. In our models, we don't usually get that many fibroblasts in the culture. I think they're pretty pure. So I've never actually used this at all, because I haven't really needed to use it.
Presumably because of the Percol.
Yeah, we use Percol, and I think the purity of the myocytes you get is quite high, but also because I guess maybe two reasons. One reason is usually I use quite a confluent monolayer. And when you do that, the fibroblasts, they can't really proliferate too much anyway. There's no space for them, so they don't proliferate. The other thing is that, so we use quite low serum concentrations in these cultures of neonatal myocytes. It's just, I think, between 1% and 5% horse serum to actually plate the cells, and then after 24 hours you change that to a 1% FBS medium for the rest of the time, or even no FBS at all. And so in that situation, the proliferation rates of fibroblasts are generally reasonably low, low enough that it hasn't really affected the model so far. So I haven't ever used this in culture.
Luke Bertrand, is there any differences in terms of metabolic pattern using your protocol? I mean, I've not come across any different. Well, we've not done anything like this, but I don't know if you have.
So by metabolic pattern, what do you think Luke is referring to?
I suspect whether we've done any analysis on, which amino acids are present, what happens to the Krebs cycle, maybe something.
You mentioned the insulin response in his question.
Possibly, yeah, maybe insulin responses, but
yeah.
I mean, we've not done it. Well, I've not seen any of that anyway. So.
No. So we use insulin in our culture media, and that's been used in myocyte culture media at least since the 1980s by the Paul Simpson group. And that improves their viability in culture. So some people have asked, is that actually having effects on things like hypertrophy? But usually I think when they take it out of the media, it hasn't had too much of an effect.
Luke has just responded underneath. Glucose, a fatty acid use, insulin response. So exactly what Nina said.
Yeah.
Okay, right, yeah. We haven't measured it. If we're comparing this to Langendorff, we haven't made the comparison, right? No. What I do know is that if we stimulate the cells with, for example, adrenergic stimulation or hypoxia, they'll have the responses that we expect. So they upregulate genes involved in glucose uptake and glucose metabolism. We haven't done detailed, for example, seahorse assays. I don't think we've done. Maybe some people in my lab have done, but I haven't done. I can't remember now. So I don't think we've done seahorse to see real metabolic data.
Yeah. May I ask, you're using the M199, right? The medium. Yeah. I think it has high glucose, around 25 millimolar. So I suspect that those cells in culture are, this is what I think, you have to test it, that they rely on glycolysis rather than on oxidative phosphorylation to produce ATP. This is what I would think.
So I can just jump in there. I say that we actually use M199 as it's not high glucose. So M199 is 1 gram per liter. It's 1 gram per liter glucose. So that's about 5 millimolar. So that's about actually normal glucose, right? Okay. So we find that if we use DMEM, which is often high glucose, which is about 4.5 grams per liter, right, about 25 millimolar, then you will start to sometimes get hypertrophic responses in cells if you do that. But to your other point, I agree. I think that in the culture, the cells, they might well be using glycolysis a lot. And certainly when you, for example, you put them in a hypoxia chamber, they'll really turn to glycolysis then. There are people trying adding certain fatty acids, things like palmitate. And we also did this a little bit as well when we added the lipid concentrate to the myocytes to see if that could help them survive longer, and I think it does. So I do think that if you add other substrates into the media, they might well use that, although it could also be acting in different ways. So for example, lipids can signal in non-metabolic ways as well, right? So we can't discount that.
Yeah. Well, believe it or not, Nina, we've reached the last question.
Have we?
There's still 100 people.
Let me turn on the light. It's dark.
It's getting light here already.
So, do you want to take this one?
Where are we? Sorry, I've lost my...
Patricia.
Okay. Is any better is adenoviral infection better than lentiviral infection? I would say both work. I think adenoviral generally gives higher infectivity than lentiviral. Yeah. We also found quite a nice way of using the adenoviruses. So you can actually, and this is at least for the neonatal cells, we can actually incubate the adenovirus with the cells for just half an hour in the incubator before you plate them. So this is just in like an Eppendorf tube, in a 1.5 ml Eppendorf tube in media. And that could be quite nice because then you can concentrate your cells in your adenovirus. Then you plate the cells the next day. For example, if that's a GFP containing virus, they'll all be green. So you get very good transduction that way. And we use polybrine it's polybrine, right? So we use polybrine for that as well.
I think,
guys, you should be commended for your resistance and kindness. And
is that all you got? Come on.
thank you for addressing more than 50 questions. You have been really remarkable, and thank you for resisting for almost 2 hours.
Thank you.
And also thanks to the audience, because I really see that we have a lot of participants still connected, and this is really fantastic. So it seems that people appreciated your presentation and your address.
Yeah.
They were curious to hear your answers, so that's fantastic.
Yeah, no, I really appreciate people coming to watch.
Yeah.
Nina, I'm always curious to hear my answers too.
Yeah, I'm not sure I want to know my answers.
I never know what's going to come out. No, thank you both. I mean, and Matt, thanks for all the work. Nina, thanks for putting up with both of us.
It was a pleasure, and I was very much interested in it because, in fact, we are also using adult cardiac myocyte cultures in the lab, so it's something that is relevant for all of us. So I think that's why of the big success of this webinar. So we see each other on Thursday, right? We said with the next speaker. I don't know if you want to put up the slide again, just to remind.
So we can just, so we take a break tomorrow, but then.
Yeah, on Thursday we have Rick Southward, so we will meet again then. Thank you, everybody, and have a nice evening and afternoon and the rest of the night for Matthew, for example.
Yeah, sleep well. You deserve it.
Way past my bedtime.
Thank you, guys.
Bye-bye.
Okay, thank you. Bye-bye.